We are going to move into our first session, which we're focusing on the endocannabinoid system, and I'm delighted to turn the podium over to Dr. David Shurtleff, who will be one of the co-moderators along with Dr. Aidan Hampson. So with that, David, may I turn it over to you?
Sure.
Okay, thank you.
Thank you, Jack, and welcome everybody. It's a great pleasure to be here. As Jack mentioned, my name is David Shurtleff. I'm the Deputy Director of the National Center for Complementary and Integrative Health, formerly known as NCAM. It actually took an act of Congress in December of 2014 to change our name, which we greatly appreciate. I'm the co-chair of the session along with my colleague Aidan Hampson, who will be introducing the other two speakers and moderating this session, and he's from the National Institute on Drug Abuse. So this morning, the three speakers for this session will give us a comprehensive overview of the fundamental biology of the endogenous cannabinoid system, including an understanding of the cannabinoid receptors CB1 and CB2 of the endogenous cannabinoids. And they will also discuss the endogenous cannabinoids themselves, such as anandamide, and how they function, and particularly for our first speaker, how they are degraded in the brain and the body. In addition, speakers in the session will help us understand how the naturally occurring so-called phytocannabinoids found in marijuana, such as delta-9 THC, work on the endogenous cannabinoid system to produce their effects in the brain and the body. The session will also begin to touch on how the endocannabinoid system might be used and modulated for treating a variety of diseases and disorders. So it is now my great pleasure to introduce our first speaker, who I've known for quite a long time and is really a leader in the field of the endogenous cannabinoid system, Dr. Benjamin Cravatt. Dr. Cravatt is a professor in the Skaggs Institute of Chemical Biology and chair of the Department of Chemical Physiology at the Scripps Research Institute in La Jolla, California. Appropriately, the title of his presentation is The Endogenous Cannabinoid System, an Introduction, which is great. Please welcome Dr. Cravatt.
Thanks, David, for that introduction, and thanks to NIDA and the other organizations for putting together this interesting meeting. So I've been tasked with trying to introduce the endocannabinoid system to you, along with my colleagues Ken Mackie and Daniele Piomelli. So, of course, the name endocannabinoid originates from the fact that there are exocannabinoids, such as the active ingredient of cannabis, tetrahydrocannabinol, or THC. THC enters the body and eventually the human brain and produces most of its neurobehavioral effects through a single G protein coupled receptor, cannabinoid receptor 1. There is a second cannabinoid receptor 2, which is also expressed in discrete regions of the brain and the immune system. But most of the effects that we associate with cannabis use, including those that are beneficial, such as improvements in pain relief, appetite, reductions in spasticity, as well as the effects that may be untoward, motor defects and cognitive impairment, occur through the central CB1 receptor. So back in the early 90s, Rafi Meshulam's lab, along with others, determined endogenous constituents that activate the CB1 receptor. These are essentially neutral lipids that represent conjugations with arachidonic acid, either conjugations with ethanolamine to create this fatty acid amide, anandamide, or conjugations with glycerol to create this fatty acid ester, 2-arachidonyl glycerol. Both anandamide and 2-AG, or 2-arachidonyl glycerol, bind the CB1 receptor with similar affinities in the mid-nanomolar range. They do have different properties of agonism, which I think Ken will get into in more detail. And so what I'm going to review for you today is trying to understand how the endocannabinoids function through the lens of trying to perturb the enzymatic processes by which they are produced and degraded. So this is just a summary of sort of the enzymatic regulation of endocannabinoids, and it's important, I think, to understand briefly why preclinical researchers tend to gravitate towards the enzymes that regulate lipids like endocannabinoids. Lipids are, of course, not directly encoded by the genome, right? They're products of metabolism, and therefore, to get genetic and pharmacological access to the endocannabinoid system, one often has to identify the proteins that regulate those signaling molecules and transmitters. And so both endocannabinoids are thought to be made postsynaptically in neurons by different enzymatic machinery, which we'll review later in a moment. And then they would then traverse backwards, retrograde fashion to the presynaptic neuron, where is the localized endocannabinoid receptor, CB1, found. producing a variety of signal transduction effects that I think ultimately culminate on outcomes such as a reduction in the probability of synaptic vesicle release. Then the endocannabinoids are terminated in their signaling by discrete enzymatic processes. And so we've been fortunate that the enzymatic pathways that regulate endocannabinoids, 2-AG and an anandamide respectively, are distinct, and it allows us to target those proteins for understanding. So I'm going to focus first on the enzymatic inactivation of endocannabinoids because I think there we have the largest body of research that's begun to inform us on the function of endocannabinoids in the nervous system. And even though the chemistry shown here of hydrolysis of the amide and the ester bond of anandamide and 2-AG respectively is similar, there are discrete enzymes that control that process to generate arachidonic acid. And arachidonic acid, even though it can have its own biological activities, does not act as a CB1 or CB2 ligand. So we'll focus first on anandamide enzymatic hydrolysis, which is primarily regulated by a single enzyme in the nervous system called fatty acid amide hydrolase, or FA. So FA is an integral membrane enzyme, probably accesses its anandamide and fatty acid amide substrates directly through the lipid bilayer, as shown here in this model, hydrolyzing the amide bond to give you the corresponding fatty acid. So when FA was characterized back in the mid to late 90s, this gave us some tools to begin to study this system. And for instance, one could create targeted disruption animal models, such as knockout mice, to study FA. And those animals were created. They grow up, they're viable, they're fertile, they're healthy. They really show no overt alterations in their cage behavior or function. But when you look in the nervous system of these animals, you see that they have highly reduced hydrolytic activity for the endocannabinoid anandamide and highly elevated concentrations of anandamide and other fatty acid amides. So this is an interesting case where even though the animals appear normal, they have a massive basal change in their neurochemistry, that being elevations in the endocannabinoid anandamide and a variety of other fatty acid amide lipids which have their own signaling functions, not through cannabinoid receptors. That inspired researchers to look at stressing these animals in different ways to see if phenotypes could be revealed. And one area where phenotypes have been consistently revealed in FA-disrupted animals is in pain models. So this is a formalin pain model of nociceptive and noxious pain, where you see that the knockout animals show reduced pain behavior. And this is reversed by a CB1 receptor antagonist, but not a CB2 receptor antagonist. So we would interpret these results to mean that the heightened endocannabinoid tone in these animals can produce an anti-hyperalgesic or an analgesic effect in these animals, and that is through activating cannabinoid 1 receptors. And again, this is against a backdrop of rather limited other overt behavioral differences in the genotypes, which inspired the development of FA inhibitors as perhaps another way to manipulate the endocannabinoid system for our knowledge gain, but also as potentially clinical opportunity. And I think Daniele will review the clinical work in more detail later. I just wanted to highlight that these come from both academics, including Daniele's lab and Dale Boger's lab, as well as companies such as Pfizer. And I wanted briefly to show you how these inhibitors work, because it provides you with an appreciation for the types of technologies that the academic research community has developed to optimize the specificity and activity of these compounds. So most of those inhibitors operate by covalently inactivating the FA enzyme. So FA is a serine hydrolytic enzyme which uses a serine nucleophile for catalysis. Those inhibitors enter the active site and they react with the serine nucleophile to form this carbamylated product, which is essentially an irreversibly inactivated enzyme. And our lab had the fortune of working with Pfizer during the development of these inhibitors. And they were obviously, as a pharmaceutical company, interested in understanding the specificity of such covalent mechanisms of action for their inhibitors prior to taking them into clinical development. And this allowed us to use some chemical proteomic technologies that our lab and others have developed to ask and answer that question. And I think it's valuable for the audience here in particular to understand how great of there have been in advances in understanding how pharmacological agents like enzyme inhibitors work in the body today with the advents in mass spectrometry-based proteomic methods as well as chemical biology methods. And so in this case, what we simply do is take a lead Pfizer inhibitor, we embed within this molecule a simple alkyne unit. This gives you a latent handle. That allows you to take that molecule, administer it to any living system you'd like, a cell, an animal model. That molecule will go and react with whatever proteins it reacts with. They become alkynylated. You can't directly visualize that, but using some advanced chemistry developed by Barry Sharpless's lab at Scripps, you can then click on a reporter tag, which allows you to then visualize those proteins that react with your small molecule. So in a nutshell, with today's technologies, you can take any small molecule and identify any protein that small molecule reacts with in any living system. So when we did that, when we took this compound with Pfizer, administered to animals, waited a certain amount of time, and then sacrificed those animals and looked at all the proteins that reacted with this compound in the rodent brain, this is the profile that you see, one protein, and that protein is lost in the FAW knockout mouse. So this was exquisite evidence that these compounds are remarkably selective for FAW over other proteins in the body, in the brain, and that was some of the evidence that eventually led to these compounds entering into human clinical development. Turning to the enzymatic hydrolysis of 2-AG, again, yielding arachidonic acid, this is primarily, not exclusively, but primarily regulated by an enzyme, monoacylglycerol lipase, in the nervous system. MAGL is not an intramembrane enzyme. It binds to the membrane through these lid domains in a peripheral manner to access 2-AG for hydrolysis. So knockout mice were also generated for MAGL to try to understand how the 2-AG signaling lipid performs, and this is just the loss of MAGL that can be visualized with chemical proteomic methods. And again, kind of analogous to what one sees in the FAW system, if you block MAGL genetically, you see massive elevations in the 2-arachidonoglycerol endocannabinoid, no changes in the anandamide lipids, so these are really orthogonal systems. But here we were met with somewhat of a surprise in that these animals showed no alterations whatsoever in their pain behavior. In fact, they really showed no evidence of any sort of cannabinoid-based behavioral effects, and that was perplexing to us at that time. Fortunately, around that same time, inhibitors against MAGL were developed. Those also nicely replicated heightened elevations of 2-AG throughout the nervous system, no changes again in the FAW substrate anandamide. And here, these inhibitors did produce reductions in pain responses in a variety of animal models. And so the question then became, why are the genetics and the pharmacology apparently inconsistent with one another here? And we heard earlier about the fact that anytime one does pharmacology, you must understand dose and duration. I would say that genetics also falls in that same category because genetics is the ultimate long duration knockout. And indeed, when we attempted to treat animals chronically with high doses of the MAGL inhibitor, we began to replicate the phenotypes observed in the MAGL knockout, that being loss of this altered reductions in pain behavior or hyperalgesia. No such change occurred with a FAW inhibitor. So this implied that prolonged blockade, at least extensive blockade of MAGL, led to tolerance in these pain assays, and that could be nicely correlated with alterations or adaptations, if you will, in the cannabinoid 1 receptor itself, whereas you saw either from the MAGL knockout or animals treated chronically with high doses of an MAGL inhibitor, reductions in the CB1 receptor content and activation state in the brains of these animals. So that is a very different outcome than what we saw in the FAW case, and we'll get back to what might be going on there in a moment. What is interesting, however, is when one looks back at the acutely treated animals where you've blocked MAGL, you not only see these alterations in pain behavior, but a variety of other behavioral effects, including impairments in motility and changes in body temperature that are reflective, if you will, of a broader, potentially broader activation of the cannabinoid system. So from the studies of enzymes that degrade endocannabinoids, I think we've learned a couple things. One, we've learned that the two endocannabinoid lipids themselves have overlapping in the sense that they both appear to regulate certain behavioral processes like pain regulation, but also distinct, divergent functions. I think it would be fair to say that most of us would agree that the 2-AG signaling pathway appears to be broader in its influence over the nervous system than the anandamide pathway. And this is an interesting feature of the endocannabinoid system that we've called ligand diversification. It's differentiated from, say, how monoamines signal, or monoamines typically signal through receptor diversification, right? So you have serotonin and many serotonergic receptors, dopamine and many dopaminergic receptors. Here you have predominantly a single G protein coupled receptor, CB1, driving most of the nervous system effects that you're observing, but then two ligands that are being regulated differentially to control the system. And I think that's an interesting twist to how this pathway signals in the brain. There are some physiological implications to the work that's been done in understanding endocannabinoid and activation pathways. We might come to the conclusion that 2-AG is more of a workhorse endocannabinoid, broadly expressed throughout the nervous system, tonically being released to regulate a variety of neuroplasticity processes, which I think Ken will review later on, and I think he'd probably agree that most of those appear to be due to 2-AG production. One might consider anandamide more of a stress-responsive endocannabinoid, which is why animals that lack FAW are relatively normal, but then reveal a variety of interesting phenotypes. I've only reviewed a subset for you here that are more related to stress and stress-responsive. Then from a translational perspective, which I think Daniele may touch on in a bit more detail, you can ask these questions. So does that mean that by elevating 2-AG through blocking its degradation, is that too much CNS pharmacology, or is that just right? On the other hand, if you elevate anandamide, is that too little CNS pharmacology, or just right? And these are the questions I think we're still grappling with in the field. And this is not an unprecedented challenge in nervous system pharmacology, right? There are many examples of direct receptor agonists that stimulate pathways, and then one tries to come at those pathways through blocking the degradation of the endogenous ligands and trying to or the uptake of those endogenous ligands and trying to understand how to produce the right amount of CNS pharmacology for therapeutic benefit while minimizing unwanted side effects. Finally, you could ask the question, if there are two different pathways, do they crosstalk in any way? So far, what I've shown you would suggest that the anandamide FAW and 2-AG MAGL system are operating in almost exclusion of one another. And that's not totally correct. If one actually does begin to experimentally ask the question, what happens when you block both these pathways, for example, by administering a MAGL inhibitor to a FAW knockout mice, you do see evidence of additivity or synergy in processes like pain regulation, but you also begin to now pick up an interesting set of side effects that resemble more direct cannabinoid receptor 1 agonists. And so in some ways, life has given us a little bit of a reprieve here by producing two different endocannabinoids and having them regulated in different ways, sort of carving up, if you will, the effects of a direct cannabinoid agonist and distributing them across two different endogenous ligands. We don't know how this crosstalk occurs, and I think this is another interesting area of future research. One could imagine that they even compete for the same receptor at the same circuits, or that anandamide and 2-AG are signaling through different circuits that then crosstalk at a more holistic level, at a systems level in the brain. And of course, these aren't mutually exclusive models, and I think Daniele and Andre Homan published work several years ago indicating and underscoring the differences in the sort of duration and magnitude of signaling of these lipids, and I think that's an interesting area of future research. So to wrap up in the last few minutes, I did want to touch base on the enzymatic production of endocannabinoids, where I think we're beginning to gain more knowledge. So 2-arachidonyl glycerol, or 2-AG, is produced through an enzymatic pathway that begins with phospholipids and travels through a phospholipase C enzyme to give you a diacylglycerol intermediate, which is then converted to 2-AG. These diacylglycerol enzymes seem to be dedicated enzymes in the brain and other tissues responsible for producing 2-AG. They are multi-pass integral membrane proteins of ill-defined structure and emerging functional understanding. I just wanted to summarize with one slide what we know in this system. So you might ask the question, why are there two different diacylglycerol lipase enzymes, alpha and beta, for regulating endocannabinoid production or 2-AG production? And we don't really understand that in detail yet, but we're beginning to get some ideas and knowledge. So diacylglycerol lipase alpha is predominantly expressed in neurons throughout the brain, with more limited expression in support cells. As a result of this expression level, if you disrupt diacylglycerol lipase alpha, you will see that you dramatically reduce 2-arachidonyl glycerol endocannabinoid content throughout the brain. And this then leads to impairments in a variety of synaptic plasticity processes, which I believe Ken will review in more detail later. So this is additional evidence to support that 2-arachidonyl glycerol is a major workhorse endocannabinoid in the CNS. In contrast, the diacylglycerol lipase beta enzyme is also expressed in the nervous system, but is mostly found in support cells such as microglia, where diacylglycerol alpha is absent. And so what this means is that even though diacylglycerol beta disrupted animals have no change in bulk 2-arachidonyl glycerol content throughout their nervous system, they actually show interesting phenotypes in neuroinflammatory models, where they show reductions in inflammatory responses, suggesting that selective perturbation of the diacylglycerol lipase beta enzyme could produce some interesting subsets of effects in the nervous system that impair inflammatory responses, say in neurodegenerative models, while sparing the brain of bulk alterations in endocannabinoid signaling that would presumably come from disrupting the diacylglycerol lipase alpha enzyme. Anandamide biosynthesis has proven to be a very complex process involving many enzymes, and I'm not going to subject you to a review of this slide in any more detail, especially this last minute or two of the talk. But just to highlight for you, this remains an ongoing area of active investigation, likely in my mind reflecting a segregation of pathways that can regulate anandamide production ultimately in different contexts and cell types. So to finish up in the last minute on key questions and future directions. So I do think that we tend, or historically have tended to view the endocannabinoid system as operating somewhat in isolation. So we've been fixated on the enzymes and the receptors and the processes by which 2-AG and anandamide signal. But as noted in this simple numeric summary, 2-AG is a much more abundant endocannabinoid at bulk levels than anandamide in the brain. And I think this then leads to some interesting crosstalk with other lipid signaling pathways. The conversion of 2-arachidiolglycerol to arachidonic acid provides substrate, for example, for that arachidonic acid to feed into prostaglandin pathways, which can then regulate a variety of neurological processes that are unrelated to direct endocannabinoid signaling. So I don't think we can consider the endocannabinoid system separate necessarily from other lipid signaling pathways in the brain. And when we perturb the proteins involved in metabolizing endocannabinoids, we may also indirectly influence those systems as well for benefit or for cost. I also think that we in the field still struggle with trying to map all the additional components in the endocannabinoid system that may play functional roles. I've reviewed for you here the major enzymes that are involved in this process. They are not the sole enzymes by any means. There are also likely transport mechanisms that help move endocannabinoids between cells, within cells, that are still emerging in their understanding. And finally, especially in the case of the FAW anandamide system, that lipid is regulated both in biosynthesis and degradation by pathways that control a whole variety of other fatty acid amide signaling lipids as well, which have their own receptors and functions, and so they get manipulated in parallel, both endogenously and through our pharmacological and genetic tools. And finally, I think as Daniele will summarize, I think later, one question that we all have at the forefront of our minds is whether the endocannabinoid system modulation can produce a clinically useful subset of the behavioral effects of direct cannabinoid receptor agonists. And I realize that most of the meeting the next day or two will be about direct cannabinoid receptor agonists, but I think it's worth keeping in mind that many of the most transformative drugs that we've used in the nervous system are not direct exogenous agonists of receptors. Rather, they are ligands that perturb the uptake, degradation, or production of endogenous transmitters. And so that's something that we in the endocannabinoid field have been especially interested in trying to develop the tools and understanding to progress that aspect of our field. With that, I will conclude. Thank you.
And now it's my pleasure to introduce Dr. Ken Mackie, who's from Indiana University, Bloomington. He's the professor of psychology and brain sciences there, and also the Linda and Jack Gill Chair and director of the Gill Center of Biomolecular Sciences. Dr. Mackie has been in the cannabinoid field for a very long time. He's started out as an electrophysiologist and is interested in both the receptors and the endocannabinoid system, but today works with a great variety of techniques, including electrophysiology, imaging, and molecular biochemistry as well. Dr. Mackie.
Thank you, Aidan, for the introduction, and glad to be here. Thanks to Nora and Jack for conceiving and implementing this two-day symposium. I think it'll be really great. I've been tasked with covering cannabinoid receptors and their pharmacology. It's a very large field. I'll have to necessarily go fairly quickly, but please drop me an email, and I'm happy to send anyone my slides that is interested in that. Okay. So I'll talk about cannabinoid receptors. Okay. Talk about cannabinoid receptors. And to my mind, there's really two reasons for studying cannabinoid receptors. One is to understand how the effects of cannabis are produced, as Ben mentioned, most of the effects of cannabis, psychoactive effects of cannabis are mediated by CB1 receptors, but also to identify potential entries into therapeutic manipulation of the endocannabinoid system. So I'll give a brief introduction to endocannabinoid receptor signaling, cover some important concepts because, as heard earlier, understanding pharmacology is really key to understanding how these drugs work. So cannabinoid receptors are proteins that interact with endogenous and exogenous cannabinoids. The best characterized cannabinoid receptors are the CB1 and CB2 cannabinoid receptors. There are additional cannabinoid receptors that I won't have a chance to talk about, and those particularly interact with endogenous cannabinoids. They include nuclear receptors such as the PPARs and TRPV1 channels. Also, THC and endogenous cannabinoids can interact with many other cellular targets, although often the biological significance of these interactions are obscure. As we heard earlier, dose really is very, very important. So I'll mostly focus on CB1 receptors, although a lot of the concepts will also carry over to CB2 receptors. CB1 and CB2 receptors are both G-protein coupled receptors, and as such they have very classic signaling pathways shown here: inhibition of adenyl cyclase, activation of various MAP kinases, inhibition of presynaptic calcium channels, activation of certain potassium channels. As far as localization goes, CB1 is primarily expressed in neurons and some glia. CB2 receptors primarily expressed in endothelium and microglia, and some possible neuronal expression. CB1 is mildly inducible. CB2 is highly, highly inducible following inflammation and injury, which has interesting therapeutic applications. I think it's important to understand where CB1 receptors are located because location really informs function. This is work from David Lewis's lab, a coronal section of monkey brain stained with antibodies against the CB1 receptor. This, for those of you who aren't neuroanatomists, this is the back of the brain. This is the front of the brain. The thing that's very striking here is cannabinoid receptors, CB1 receptors are widely and highly expressed. They tend to show up in a laminar pattern in many areas of cortex. In cortical areas, they're most highly expressed in association areas, less highly expressed in primary somatosensory and motor areas. There's subcortical expression, particularly striking in basal ganglia. We have very, very high levels in globus pallidus, and also pretty high levels in some nuclei of the amygdala. CB1 receptors are relatively sparse in white matter and low but not absent in thalamus. So a very interesting distribution. If you look at where message for mRNA for the receptors expressed, you come up with a pattern. This is sort of recapitulated throughout cortex. Hippocampus, of course, is shown here. And what you immediately see is you have a few neurons that have very, very high levels of expression, shown here as intense blue, and then more neurons that have a much more moderate level of expression, shown here in light blue. At the protein level, this translates into this kind of pattern here. Again, pretty high levels found in what are, those of you who know hippocampal anatomy, are synaptic areas of hippocampus. But the receptors expressed here are important as well. From using mice lacking receptors in certain types of neurons, you can get quite a bit of information about where these neurons are located on the cellular level. So this is wild-type mouse here, with, again, CB1 receptors in brown. For comparison, staining for CB1 receptors in a CB1 receptor knockout mice, see no staining. If you knock out CB1 receptors from GABAergic neurons, just leaving CB1 receptors present in glutamatergic neurons, you see very, very low levels of expression, but in distinct areas here, terminals of hippocampal mossy cells, which are quite important in epilepsy, as we'll hear later from Yvonne. Although these receptors are very low in number, they function differentially, and this just shows a measure of receptor activation using G-protein coupling. This is for normalized signaling in wild-type neurons at 1. If you knock out CB1 receptors from GABAergic neurons, primarily you're left with CB1 receptors in glutamatergic neurons, and those signal very efficiently, whereas the CB1 receptors in GABAergic neurons signal less efficiently, so there's about a 5-to-1 ratio here. And there's a large variety of experiments done with mice lacking CB1 receptors in GABAergic neurons but present in glutamatergic neurons. showing that CB1 receptors in those glutamatergic neurons have very major biological effects. Okay, if we zoom down in terms of level of analysis, we can look here in hippocampus at light microscopic level. You see this intense staining pattern for CB1 receptors. This is typical of receptors expressed on axons. And then at the electron microscopic level, shown here, you can see this is a GABAergic terminal where CB1 receptors are detected with immunogold. It's these little clumps here. See quite high levels of expression, and then occasional expression on glutamatergic terminals. And you also note that glutamatergic terminals having CB1 tend to make synapses on the spines, whereas GABAergic terminals tend to make synapses on the shafts. So the localization of CB1 receptors and how they signal suggests they may inhibit synaptic transmission, and that's definitely the case. And I want to show you briefly kind of the type of experiments that demonstrate that. So this is recording from a hippocampal slice, where you record from a pyramidal neuron here, you stimulate GABAergic fibers out here, and this is stained with CB1 receptor antibodies to illustrate how densely CB1 receptors express. And this is the kind of experiment results you get. So this is a iPSC evoked by release of GABA. You can see stable levels here. If you put on this CB1 receptor agonist, WIN, you see a diminution of the amplitude of the iPSC, and that is reversed by a CB1 receptor antagonist SR. So this is kind of classic CB1 receptors inhibit synaptic transmission. So what is one of their endogenous functions? So I've shown you that Ben's shown you that neuronal activation can produce endocannabinoids. These activate CB1 receptors to inhibit synaptic transmission or suppress neuronal excitability. There are 4 major forms. This is a type of synaptic plasticity that's really widespread throughout the brain. 2-AG is often, but not always, the endocannabinoid involved, and the effects can either be transient or long-lasting. I'll take a couple of minutes to run through some of those. So the first is DSI, DSC depolarization-induced suppression of inhibition excitation here. Strong depolarization of a neuron inhibits subsequent inhibitory input onto that neuron. So this is recording from neuron layer 5 of cortex, and just spontaneous inhibitory transmission onto that neuron. You depolarize the neuron, and that shuts off transmission onto that neuron for a few tens of seconds. And the schematic that's going on is like this. So depolarization of postsynaptic neuron increases intracellular calcium, leading to the synthesis of endocannabinoids, 2-AG primarily, diffuses retrogradely, activates cannabinoid receptors to inhibit the probability of release. And this also occurs at excitatory synapses where it's called DSC. So this is another way of conceptualizing DSC. So here we're periodically stimulating the neuron. Each stimulation evokes EPSC. If at this point we depolarize the neuron, we see a diminution of the evoked responses. And that can be quantified in a dose-response curve where on the x-axis, increase in duration of depolarization increases the suppression of the EPSC. So we can analyze this using typical pharmacological approaches. So this I showed you with DSC. Another form is MSC, where instead of depolarization, you have activation of metabotropic receptors such as mGluR5 or mGluR1 or M1, M3 receptors that can also produce endocannabinoids that inhibit synaptic transmission. Like DSC, this is a brief process. However, if this glutamate, in this case, has been release has been going on for a long time, you transform from a short-term inhibition to a long-term inhibition or a type of LTD, where if we look on the purple neuron here, first, this mGluR5 receptor is stimulated for a long time, produces endocannabinoids that set in process a long duration action that leads to long-term depression of this synapse. However, you can also have, and this is homosynaptic, you can also have heterosynaptic LTD where these endocannabinoids are produced because of glutamate released by this neuron, but act on cannabinoid receptors on another neuron, say a GABAergic neuron, and get long-lasting inhibition of this terminal here. So inhibition of a glutamatergic terminal, long-term depression will decrease excitability. A long-term depression of an inhibitory neuron will actually increase excitability. This just illustrates that the net effects of endocannabinoid-mediated synaptic plasticity can be quite complex and are very context-specific. The final form of endocannabinoid plasticity is quite different. This is cell autonomous. So this is slow self-inhibition where you have in a small number of neurons, repetitive depolarization, increases intracellular calcium, activates endocannabinoid production, stimulates CB1 receptors, leading to activation of intrarectally rectifying potassium channels and prolonged hyperpolarization. So this is a way of quieting down a single neuron. All these are kind of transsynaptic events. Okay. So CB2 receptors, wanted to briefly mention these. These are most abundant in immune cells, including microglia. They're highly inducible, may be expressed in some neurons. They're possible mediators of some of the anti-inflammatory actions of cannabinoids. There are a number of intriguing studies that suggest that CB2 may be actually an interesting therapeutic target with minimal psychoactivity that you normally would have with CB1 ligands. I don't have time to talk about those, but there are a number of posters over the next 2 days that cover that material, and I really encourage you to go look at them if you're interested. I'd like now to move to some important pharmacology concepts that I think are really key for understanding how THC and endocannabinoids may function. The first is efficacy, the second is functional selectivity, and the third is allosteric modulators. So efficacy is a pharmacological term that just means the maximal effect. And efficacy can be positive, as it would be with an agonist. It can be zero, as with a neutral antagonist, or negative, as it with an inverse agonist. And that's just shown here, an experiment from several years ago looking at one measure of signaling, here GDP-MS binding, where you can see the agonists WIN and CP increase GDP-MS binding, and the inverse agonist SR, this is rimonabant, decreases GDP-MS binding. So efficacy can go either way. Inverse agonism, shown here, requires a fraction of the receptors to be in active conformation, and that's particularly likely to happen when you have high levels of receptor expression, as happens in the CB cannabinoid system. Although of note, it can be challenging to distinguish inverse agonism from antagonism due to ongoing endocannabinoid release, and that's something that we always worry about. Another important concept is that maximal response varies by ligand. You can have ligands of intermediate efficacy, and those often show partial agonism, which I think probably everyone's familiar with. This is an example looking at potassium channel activation by CB1 receptors. And here the data is plotted as the percent maximal response to WIN. And you can see that 2-AG is as efficacious as WIN is, which is a pretty efficacious compound in this system. However, methanandamide, a stable, metabolically stable analog of anandamide, and THC are low efficacy agonists. And conditions that affect partial agonism are low receptor density or weak receptor effector coupling. So you have to kind of know how many receptors you have and how well they couple to really tease out this aspect of pharmacology. And here's a practical example of why I think efficacy matters. Synthetic compounds such as spice compounds and 2-AG are high efficacy agonists. THC is a low efficacy agonist. And here's an example showing that JWH18, which is an early spice compound, potently inhibits EPSCs. You can see increasing concentration of JWH18 inhibits EPSCs by about 50%. That's antagonized by rimonabant, so it's CB1 mediated, and it's absent in CB1 receptor knockout mice, so that's CB1 mediated. However, if you look in this same system, so these are cultured hippocampal neurons, THC over a wide range of concentrations does not inhibit EPSCs. So in this system, THC actually acts as a neutral antagonist. However, if you do our DSE experiment where increasing durations of depolarization progressively decrease the EPSC, you see if you do the same experiment in the presence of THC, the DSE dose response curve is shifted to the right, as would be expected for a competitive antagonist. So this doesn't happen throughout the brain. It happens under specific conditions of receptor number and density, but it stresses the concept that it's really important to understand that THC may work differentially at different synapses. The second concept is functional selectivity, and here's the idea that different agonists engaging CB1 or CB2 receptors activate different suites of intracellular signaling molecules. So this is an example pulled from CB2 literature, where this compound inhibits adenyl cyclase, recruits beta-arrestin, activates ERK kinases. However, CP5594, working through the same receptor, does these things, but also stimulates receptor internalization and inhibits calcium channels. So two different receptors, both considered agonists, can actually activate overlapping suites of signaling pathways. And this is another example of why this kind of thing matters. This is looking at cell death in a Huntington's cell model line. So these are essentially indirect pathway medium-spiny neurons cultured from a mouse that carries a poly-Q expansion for Huntington gene. And you can see that, and this is measuring cell death on the x-axis, on the y-axis, so higher number means more dead cells. You can see that THC and CP both increase cell death. However, a number of other compounds, including 2-AG and anandamide, decrease cell death, all working through the same receptor. This is work from Eileen Donovan-Wright's lab, and when they looked at signaling pathways involved here, and this is a bit of a complicated slide, but the main thing to note is that you can either go up or down. Up indicates a bias towards ERK signaling, down bias towards arrestin signaling, and you can see, interestingly, that THC and CP favor arrestin signaling. This seems to culminate in cell death. Anandamide tends to favor ERK signaling, and that culminates in cell survival. And very interestingly, if you combine THC and CBD, you favor ERK signaling and you favor cell survival. So it's kind of an interesting outcome. So THC, you can't conflate effects of THC with the effects of 2-AG or other compounds. Finally, talk about allosteric modulators. So drugs can intercept with receptors at multiple sites. The orthosteric site is a typical binding site where THC binds to CB1, et cetera. Allosteric would be another site on the receptor that a ligand can interact with. Allosteric modulators may alter the efficiency of binding or the efficiency of signaling in the simplest iteration. And these interactions may be positive, so positive allosteric modulator or PAM, or negative allosteric modulator. And they're often, to make it even more complicated, they're often selected for the orthosteric ligand, a phenomenon we call probe dependence. So you may have an allosteric modulator for THC, but it's not an allosteric modulator for 2-AG. And of course, in the optimistic sense, this enriches the repertoire of drugs modulating receptor signaling. It also makes things a bit more confusing. And schematically, this is what allosteric modulator looks like. So normally your agonist interacts with the CB1 or CB2 receptor here and stimulates signaling through the G proteins or arrestins. What an allosteric modulator will do will either enhance the binding of this agonist to the receptor usually by either speeding up or slowing down the dissociation from the receptor, or it may favor receptor conformation that enhances signaling down this way for the allosteric modulator. So there's been in the last few years, a number of interesting allosteric modulators examined for cannabinoid receptors. One is particularly interesting is pregnenolone, which acts as a negative allosteric modulator, and that's shown here for one signaling through CB1 receptor. So increasing concentrations of THC stimulate MAP kinase or ERK1/2 phosphorylation, and you can see increasing concentrations of pregnenolone decrease that signaling in a non-competitive fashion. So this is kind of classic for allosteric modulator. And that has functional effects, a number of functional effects is explored in this paper. One of which I think is particularly interesting is that normally if you give THC, you get a surge of dopamine, and that surge of dopamine is strongly attenuated if you pretreat the animal with pregnenolone. Another interesting example of allosteric modulator is cannabidiol. Cannabidiols we'll hear a lot more about. It's a very fascinating molecule, has lots of targets, one of which seems to be CB1 as a negative allosteric modulator. This is another experiment looking at ERK activation by increasing concentrations of 2-AG, and you can see that increasing concentrations of cannabidiol or CBD progressively decrease the activation here. Okay, with that I'd like to wrap up with my summary slide. Sort of the things I think we know pretty well are that CB1 mediates most of THC's psychoactivity. Endocannabinoids play a very prominent role in modulating synaptic plasticity, and there's a rich and complex pharmacology of cannabinoid receptors, which I think we really need to understand how THC produces effects as well as for drug development causes. The key things I think that we don't know but would be good to know are what other receptors and proteins interact with cannabinoid receptors, on how do these influence signaling, such as accessory proteins, such as CRIP or SGIP1, or heterodimerization with other GPCRs. What are the therapeutic opportunities? Which CB1 receptors being imaged in vivo? Remember, I showed you bulk of the CB1 receptors are inhibitory neurons. Those are the ones that we see in most imaging studies. However, the low fraction that are in excitatory neurons or cholinergic neurons are incredibly important functionally, but we don't see those in our imaging studies. And then also there's a very interesting biology of CB1 receptors on astrocytes and mitochondria, which is still being sorted out. But thanks for your attention.
And finally, for the endocannabinoid section, I'd like to introduce Dr. Daniele Piomelli, who's going to discuss the therapeutic potential of the endocannabinoid system. Danielle's coming from the University of California, Irvine, where he's the professor of anatomy, pharmacology, and biochemistry, and he is the Louise Turner Arnold Chair at that department. Dr. Piomelli has been interested in arachidonic acid for a lot longer than cannabinoids were around, endocannabinoids were around. He was working on icosanoids before anandamide was discovered, but jumped in completely into the FAR when FAR was discovered, and has done a great deal of work on FAR, and has also done a lot of work commercializing FAR for therapeutic potential. He has 25 patents and 3 startup companies. So I'd like to introduce Dr. Piomelli.
Good morning. I'm honored to be here. As our thoughts go to the victims of the terrorist attack, I'd like to remind ourselves that we can do good things, and evidence-based medicine is one such thing. I've been charged with the task of going over the therapeutic potential of the endocannabinoid and the cannabinoid system at large. You have heard from the previous speakers that the endocannabinoids can regulate a variety of fundamental physiological functions. The speakers that preceded me have focused their attention on the brain, but really the entire body is a target for the endocannabinoids. I have divided my short presentation into 2 blocks. The first one will cover the cannabinoid receptors you have heard a lot already about from Dr. Mackie. And the second one will cover the 2 main endocannabinoids that were introduced to you by Dr. Cravatt, anandamide and 2-AG. So let's start off with the cannabinoid receptors. These, as we have seen, are G-protein coupled receptors, and they're present in 2 established subtypes, CB1 and CB2. They activate a rich repertoire of intracellular transduction mechanisms Dr. Mackie alluded to. The CB1 receptor is certainly one that we are most interested in. Dr. Mackie showed us some very beautiful pictures of its localization in the brain. I would like to stress, however, that this receptor is truly ubiquitous in the sense that it is expressed by many different cell types in the body, not just by neurons, not just by astrocytes or microglia, but also by hepatocytes, by adipocytes, and a variety of other cell lineages. The CB2 receptor, on the other hand, is a little bit different because it is mainly expressed by cells of the immune lineage, either innate or adaptive immunity, including macrophages, B lymphocytes. So in a sense, CB2 receptors also are ubiquitous insofar as macrophages are ubiquitous. But they are ubiquitous in a different way as the CB1 receptor is. Let's start with the CB1 receptor, the one we actually know the most about. And what can we do with this receptor? Well, we can do a number of different things, but the first one is clearly to try and activate it and see what therapeutic benefit we could achieve that way. And this is done with what in pharmacological lingo is called a receptor agonist. Now, receptor agonist is a broad term that encompasses different animals, really, quite different animals. There are such things as partial agonists, and Dr. Mackie gave us a wonderful explanation about how a partial agonist work. But let me just go over this very quickly again. It is an important concept. So if one looks at the effect size of an agonist, so one sets that a receptor can produce a response that is 100%, then a partial agonist would produce a 60% or a 70% or a 20% of their response. And this differentiates, therefore, a partial agonist from a full agonist. The full agonist will produce that maximal effect I mentioned before. So it is interesting that the two endogenous cannabinoids, anandamide and 2-AG, are different in that anandamide is a partial agonist, whereas 2-AG is a full agonist. It is also, I think, interesting that THC, the main psychoactive component of cannabis, is also a partial agonist. Now, I like to introduce you a new term, which is not really new, but I didn't make this up, but it's a term that I think we should keep in mind, a superagonist. Compounds such as CP55940 in given situations can actually cause a greater than maximal effect. So it becomes kind of a relative, saying what is full and what is super becomes almost like a relative term. But to give you a sense again that if we create probes, we can really change the response in ways that nature did not anticipate. And this is what we have done, I think, with a lot of these cannabinoid agonists, activating CB1 receptors a lot more than anandamide or 2-AG were meant to do by evolution. Finally, but not unimportantly, we can modulate the receptors through allosteric means. And again, Ken has given you a very nice introduction to this. An allosteric modulator really has little or no effect by itself, as you can see here. But if now a ligand, and for example, an endogenous ligand comes into action, then the actions of this ligand will be enhanced by the allosteric modulator. So all these things are not theory, all these things have been done. So what have we learned by using in the lab, in animal models, in vitro models, what have we learned from using these different type of receptor agonists? Well, of course, we have learned a lot about THC. There are decades of work on THC, which is a partial agonist. We know that THC is already a therapeutic agent. It is sold as Marinol, but it's also present in cannabis, and it's probably the most important cannabinoid present in cannabis. The commercial cannabis that is found in the streets or in many stores dispensaries, is mostly composed of THC at pretty high concentrations. So it is a therapeutic agent already. It does produce CNS-based side effects. And this is due to the fact that the cannabinoid receptors, or CB1 receptors, are really widespread throughout the brain. So some of the things it does, for example, we have heard the analgesic effect, we have heard about the appetite-stimulating effect, the spasticity effects. Those are useful effects, but they are inevitably accompanied by psychoactivity, by loss of memory, by loss of motor coordination. It is the nature of this drug to produce those effects. The two things cannot be separated using these type of drugs. If we look at full agonist or superagonist, this problem is magnified. These compounds are really hard to work with. They do have very interesting pharmacological actions, but they also inevitably activate all cannabinoid receptors and cause CNS-based side effects. Where we have more leeway is in the area of allosteric modulation, and this is an area that requires more work. But it's a promising area because, again, when we modulate allosterically a receptor, we are only making this receptor more sensitive to the endogenous ligand. So you have to picture this. You're not activating receptors that are not normally engaged. You're only activating, if you wish, enhancing the activation of those receptors that normally see their ligand. So you're essentially magnifying an endogenous action. That is why I believe this area is very promising and needs to be explored. Now, because it is so hard to work with direct-acting cannabinoid receptor agonists, CB1 receptor agonists, people have started to play around with an alternative possibility to make drugs that do not enter the CNS. Since the majority of the side effects of CB1 activation are in the brain, a logical way of preventing that is just making compounds that don't pass the blood-brain barrier, that act exclusively in the periphery. And there are companies and scientific and research labs that have worked on this. The results have been, however, disappointing because at least with the few drugs that have been explored, these drugs have shown peripheral side effects mediated by the cannabinoid receptor itself. As I said before, CB1 receptors are ubiquitous, and they're involved in the regulation of metabolism, particularly lipid metabolism, as well as feeding. So one common side effect of peripheral agonists has been the stimulation of feeding, the increase in body weight, as well as these metabolisms that create problems over a long period of time. However, I would like to say that this is the jury is still out there when it comes to the side effects produced by these compounds because we don't have enough of a body of knowledge yet. So that's an area that I think still deserves consideration. When we look at CB1 receptors, and agonists at CB1 receptors, we must consider the dark side of CB1 pharmacology, which is particularly represented by the superagonists that compose such abused substances as spice, K2, etc. These are compounds that typically overly activate CB1 receptors, and of course they are being now abused by a substantial number of people, particularly our youth. About 6% of 12th graders in 2014 used spice or spice-like compounds, and their impact cannot be underestimated. So what about blocking the cannabinoid receptor? Clearly we can do this, and we do this with what pharmacologists call antagonists. But again, just like agonists, they come in multiple flavors. There are neutral antagonists, there are inverse agonists, and they are allosteric or biased modulators. So what are these things? Well, neutral antagonists just block the receptor, easy. Inverse agonists seem to do something different. As Dr. Mackie explained, they actually force the receptor into a conformation that is negative, so the receptor becomes actively inactive. The allosteric or biased modulators are very interesting compounds, and again, Dr. Mackie explained them. Just let me give you again the gist of what they do. If, say, CB1 receptor is coupled to two signaling pathways, 1 and 2, okay? There are more than that, but let's say it's only 2. Well, what a biased modulator would do, typically, is just silence out one of those pathways. And if we are lucky and that particular pathway is the bad pathway and the other one is the good pathway, then we got potentially a drug. So what have we learned by using these different compounds? Well, we have learned a lot, but I think there is still a lot to be learned. We have used inverse agonists in the clinic. They've been extensively studied in animal models and then in people, particularly for metabolic diseases and obesity at the center of those diseases. We know that these compounds are effective in obesity. We know that they decrease body weight, they improve the overall metabolic panel, and they do so in a quite effective way. However, we also found that they exert CNS side effects. These CNS side effects are linked to the important role that the endocannabinoids play in regulating anxiety, in regulating mood and affect. Particularly anandamide has been involved in this in such role, and blockade of anandamide is responsible, I believe, for most of the CNS side effects of these compounds. What about neutral antagonists? Neutral antagonists are, of course, more moderate in their action because they do not force the receptor into an inactive state. They kind of block the receptor prevent the receptor from being activated. And there are studies ongoing on neutral antagonists, but we still have preciously few such compounds, and more work is needed there. What about allosteric and biased modulators? Again, here we have very promising data, and Dr. Mackie alluded to a compound, pregnenolone, which has been now optimized for the treatment of cannabis addiction. A few important thoughts about what we have learned from CB1 antagonists in metabolic syndrome. I would like to point out that therapeutic effects in human metabolic syndromes were predicted by animal studies, and the side effects also were predicted. And this is important because it gives us confidence of the predictive value of our models, so that as we test new drugs, we should believe in what we see and not just be critically skeptical. One such drug which I think deserves attention is peripheral CB1 antagonists, which have been shown to be highly effective in animal models of metabolic disease and deserve further study in humans as well. The CB2 receptor, with which I move now, is, to paraphrase Oscar Wilde, a sphinx wrapped in a mystery. We have seen that they are highly expressed in immune cells. Their function, however, are unknown. Activation of this receptor is analgesic, anti-inflammatory, and tissue protective, but the mechanism of action is unclear. But most importantly, we do not have good probes to study this receptor, and therefore more work is needed before we can definitely describe their actions. So in the next couple of minutes, I'd like to go over anandamide and 2-AG. To start off, I'd like to give you a sense of what personally I think these compounds do based on an assessment of the literature over the last several years. 2-AG, we have heard, is the workhorse. Dr. Cravatt used a very nice metaphor. Indeed it is. Another way of looking at it is that it's a point-to-point messenger. It goes from one side of the synapse to the other retrogradely. Instead of going from pre to post, it goes from post to pre, and exerts many functions in the CNS as well as the periphery. Anandamide instead is more of a modulatory transmitter. It acts like what people like to call a volume transmitter, and has responses, an array of responses that are linked to the control of stress in the central nervous system and pain in the periphery. So how can we use these signals for therapy? Well, let me remind you, first of all, how these compounds are naturally produced. They're produced from lipid precursor in cell membranes. Dr. Cravatt showed some structures. I decided that I wouldn't show you any structures, but the idea is the same. We got the lipid precursor converted into biologically active endocannabinoids, and then metabolites. Metabolites that can be active or inactive, but be what it may, if we block the biological transformation of these endocannabinoids, then we can enhance the system's intrinsic regulatory functions, possibly with greater selectivity than just activating cannabinoid receptors indiscriminately. This can be done by using inhibitors of 2-AG degradation, which target three main enzymes. They're called mannose-glycerol lipase, Dr. Cravatt described it, ABHD6 and COX-2. And these compounds have been shown in animal models so far to do a number of interesting things, which are listed here. But these compounds also have non-endocannabinoid functions, which include cancer suppression, reduced neuroinflammation, etc. Relatively mild side effects. The inhibitor of anandamide degradation prevalently target the enzyme FA, fatty acid hydroxylase, and are effective in a number of animal models, but especially in human cannabis abuse, has been shown recently, and they have a very mild side effect profile in animals and humans. I would like to point out that we know a lot about the safety of these compounds. 7 FA inhibitors have been tested, 18 phase 1, phase 2 studies have been conducted, over 1,000 subjects have been seen, no serious side effects have been noted so far. There has been recently a very tragic event in France about a compound called BA 102474. And what do we know about this compound that can explain its unusual toxicity? I don't have the time to really go over this carefully. I'd like just to point out that it's a weak Phi inhibitor, it's got an unusual chemical structure, it's a poorly characterized target selectivity, very limited animal pharmacology, animal toxicology showing lethal effects at high doses, and an unfavorable human pharmacology and human pharmacokinetics, with toxicity occurring in humans at doses that are consistent with enough target effect. So what we conclude about these terrible events, that we must shed light on many aspects of the actions of this compound, but the toxicity was most likely due to enough target effect that it was not mediated by Pha, and that much greater caution should have been exerted before and during the clinical testing for this compound. And I'd like to remind all of us that guilt by association is not sound legal doctrine. Therefore, we should not stop research on Phi inhibitors just because the one compound turned out to be a terrible molecule. So looking forward, what direction should we go to? Certainly, I believe allosteric positive CB1 modulators have offer a promise. Neutral CB1 antagonists also offer promise. Likewise, peripherally restricted CB1 antagonists, particularly in obesity and metabolic syndrome, offer promise. And both global and peripheral Phi inhibitors offer promise. Global inhibitors for anxiety, post-traumatic stress disorder, autism spectrum disorder, and a peripheral Phi inhibitor for nociceptive pain, for reasons that I didn't have the time to go through today with you, but there are animal data supporting it. Also, MGL inhibitors, or MAGL, as other people like to call it, and ABHD6 inhibitors, which target the 2AG degradation, also have promise, particularly in epilepsy, pain, and cancer. Last but not least, I'd like to mention compounds that selectively inhibit the conversion of endocannabinoids to metabolites via the COX pathway. Again, I didn't have the time to go over them, but these are interesting molecules. And with this, I am done. The light is red, and I thank you for your attention.
Okay, thank you to all of our presenters. We have, thank you for being so timely as well. We have ample time for our Q&A session, which is about to begin. We have staff wandering around both down here as well as upstairs, collecting any of your questions, so please fill those out as quickly as possible. We'll filter them to the front. And if I could ask our speakers to have a seat at the dais. And I believe, Aidan, will you be facilitating? You may want to stand at the microphone, at the podium, just for ease in that. And as we're collecting, we also are going to have some Twitter questions. We have a few already, but Aidan, did you want to just kick things off with anything before we collect some of the questions?
Well, we have a question already, which is an interesting question, and it's a question for Dr. Cravatt. Endogenous cannabinoid release, is it affected by either acute or chronic THC administration?
Not to my knowledge, but I'll let Ken and Daniele comment on that. Are you aware of any evidence that endocannabinoid production is affected by exogenous cannabinoid uptake, which I believe was the question?
Do exogenous cannabinoids affect the release of endocannabinoids?
Not that I'm aware of.
This is a very, very important area of research because the endocannabinoid system is a very plastic system. It tends to respond, as Ken has argued, tends to respond to exogenous stimuli, particularly activation of the system itself, and is very possible that the system would be engaged by exogenous drugs and respond to that engagement with adaptations. There could be persistent adaptations. We do not have data, except for a very, very few cases. For example, in exposure of adolescent animals to THC causes changes in anandamide production in the prefrontal cortex in those animals later on in life. So there seemed to be a regulation, but I think a lot more work is needed to unravel it.
I think another important aspect of that might be worthwhile discussing is while they may or may not affect endocannabinoid release, what about on the other side of the synapse? Do do exogenous cannabinoids like THC affect the amount of CB1 receptor expressed?
Very strongly. So that's very, very clear. Yeah.
And in what way does it do that? What does it do to them?
Well, you'll tend to either downregulate number or downregulate coupling, so basically make them signal less efficiently.
So essentially the endocannabinoid function would be diminished
Correct.
because the receptors are diminished rather than acting on release of the agents.
Right.
Okay. Okay, how does the distribution of CB1 receptor in the brain differ in early prenatal development versus in adult? What's in the literature?
I guess that's for me. So actually, and we'll hear much more about this later in the symposium, cannabinoid receptors, CB1 receptors particularly play a very interesting role in neural development, and they undergo very interesting shifts. So in embryonic development, in focus on cortex, you have development, you have neurogenesis, you have division, you have migration, then you have process development, synapse specification. So during synapse specification of glutamatergic projection neurons particularly, you have CB1 receptors and DAG lipase very involved in the extension of those processes and formation of synapses. So normally, as all of us has said, that in adults, you have postsynaptic production of endocannabinoids acting on presynaptic receptors. During development, the whole endocannabinoid system seems to be intact in the axon, involved in axon elongation and finding the appropriate target. So CB1 receptors have a very interesting role in development, and they undergo significant changes migrating from endocannabinoid production presynaptically to endocannabinoid production postsynaptically after development is maintained, as one example.
If I may add one more thought to this, in adolescence, again, there is clearly an increase in the number of cannabinoid receptors, at least in the prefrontal cortex, as well as the coupling to GI, along with an increase in anandamide. So this is quite important because there is data out there suggesting that the endocannabinoid system is crucial to key aspect of adolescent behavior, particularly risk-taking, which is, of course, cognate to drug addiction, but also things that are like social behavior, which are, of course, some of the things that we associate typically with the behavior of an adolescent. Likewise, the endocannabinoid system seems to sort of also change in old age, where other forms of plasticity occur, and the system also appears to be sort of monitoring these changes and changing in parallel with the rest of the brain.
Can I ask a question just about the prenatal aspect with exogenous cannabinoids? Does it change the sort of migration of those neurons early in development? Is there any change associated with exogenous availability or with exogenous cannabinoids in the system at early development?
Yeah, I mean, there's a lot of animal data on that. It's a little bit clouded because a lot of those studies have used high efficacy agonists, which don't really mimic the human situation, but the effects are widespread. If you go to the human epidemiological literature, there's certainly 2 studies that suggest there are enduring consequences of prenatal use, and they tend to focus on prefrontal cortex abnormalities.
Does anyone know whether in endothelial cells, the cells that line blood vessels, whether CV2 is constitutively expressed or whether it's inducibly expressed?
So in cortex, there's good RNA-seq data showing that you have CV2, at least mRNA, in cerebral endothelial cells under basal conditions. So it's there even under non-stimulated conditions, non-inflammatory.
But that's mRNA, not protein?
That's mRNA. Antibodies aren't terribly good, which many we can talk about coffee break.
Okay, and a question about cannabidiol. It's a question as to whether it's an inhibitor of MAGL and FAR, and could you, when discussing this, could you address whether it's an inhibitor at concentrations which are ever achieved either during marijuana ingestion or even or potentially under pharmacological conditions?
I'll take this one. Cannabidiol is the epitome of polypharmacy. It does everything, okay? Which means that it does nothing, or we are really wrong as to what it does. But it does do, among the many things that it does block anandamide transport into cells, and it does block FAR, but at high micromolar concentrations. I'm talking about 100, 150 micromolar EC50s. Now, this said, in the one clinical trial that I am aware of where these things have been tested, when CBD was given to patients, the levels, this was psychotic individuals, subjects with schizophrenia, the levels of FAR substrates were measured in those patients, and they were found to increase with the administration of cannabidiol, and also to be positively correlated with symptom relief. So if there is no direct, and those experiments suggest that there is some degree of FAR inhibition occurring because all FAR substrates in plasma of those subjects were increased by treatment with cannabidiol, and also suggested that blockade of FAR could have therapeutic utility. But as I said, CBD is the epitome of polypharmacy, so I think that we cannot draw such a simple conclusion just based on that one piece of data.
Right. Question for Dr. Mackie. Is there any evidence that THC and/or CBD acts as an allosteric orthosteric pair at the receptor, I presume, to reverse cell death compared with THC alone? Does CBD act as an allosteric modulator at CB1?
Yeah. So hard to say. I mean, because the data I showed, that's again a recent paper from Eileen Donovan Wright's lab. The evidence is all correlative. You know, you have cell death and you have these differences in allosteric modulator and signaling, but whether one's causing the other is not determined. If that answers the question. It's basically very recent work which really needs to be explored further, I think is the safest thing to say.
The next one is a very important question, but I don't know whether it's going to be something that can be answered quickly, but here's a challenge for and it's a question of the endocannabinoid system as a homeostatic system. Can you talk about it being a regulator of regulators?
I might as well start. And please, interject as you see fit. This is it's almost a philosophical question, but I think it's a very good question, as a matter of fact, because I personally believe that the endocannabinoid system is indeed a homeostatic system, homeostatic and allostatic. That is, under normal circumstances, in a healthy individual, the system can play an important role in controlling, if you're talking about 2AG, synaptic function, or if you're talking about an anandamide, the response to stress. But if the system becomes pathological, so for example, after prolonged exposure to THC, I think that it can become also allostatic. So it's homeostasis that is no longer adaptive, is maladaptive. And that is something that we always need to keep in mind, that something that is meant to regulate homeostasis can play a role also in allostasis.
And another question for Dr. Piomelli. Do COX-1 inhibitors, these are drugs such as non-steroidal anti-inflammatory drugs, aspirin and ibuprofen, do COX-1 inhibitors have any effects on 2AG or anandamide levels?
Yeah, okay. Hopefully won't reverberate. Do COX inhibitors have any effect on 2AG and anandamide? COX-2 inhibitors do. COX-1 inhibitors don't. The COX-2 is, first of all, COX stands for cyclooxygenase, which is an enzyme that makes prostaglandins. And utilizes arachidonic acid predominantly as a precursor. However, endocannabinoids can be also used as precursors, but only by the 2-isoform of cyclooxygenase COX-2. The one isoform has a very narrow substrate binding domain that doesn't allow the entry of anything else but arachidonic acid. Some mixed COX-1, COX-2 inhibitors, such as fulbiprofen, for example, ibuprofen to a certain extent at high doses, but most importantly carprofen, which is not used in people, they will block COX-2 and Phi at the same time because of this similarity. And so they will affect the levels of endocannabinoids through that pathway by blocking Phi.
Right. Okay. So here's a question about THC as a partial agonist. We know it's been discussed that it's a partial agonist, not a superagonist. If you take enough THC, does the high concentration make it become a superagonist, or would its effects be? Could it have an effect that's so long-lasting that it has downstream effects that would be like a superagonist?
To answer the first part of the question, no, that's kind of the definition of a partial agonist. With a given receptor density, you'll only get so much effect. But of course, that's looking at a simple dose response. When you look at the whole animal and you give a huge amount of drug and it lasts a long time, there's a lot of other things that goes on. So, you'll basically get long-lasting effects, very stoned, but it will still be qualitatively a little bit different from taking a spice compound for a very long time.
Yeah, I'm sorry, just if I may interject. I think it's really important to understand this concept because this is a crucial concept in cannabinoid pharmacology, and I'm so glad that Ken developed it so well because it's really very, very important. But a partial agonist is a partial agonist is a partial agonist. Yeah. So THC will not do what CP55940 does. That's about it. But then if you take a whole lot of THC over time, stuff can happen, but it's not the same.
If you combine the two, the superagonist with the partial, is there any additive effect here, or how does that? Antagonistic.
Yeah,
okay.
it can actually antagonize. Yeah.
THC would actually is the superagonist.
Yeah, potentially.
So FAR inhibitors have at least preliminarily been shown to reduce cannabis withdrawal in one human study. It's still preliminary data, but it looks interesting. Somebody's asking whether MAG lipase inhibitors might also be used to reduce cannabis withdrawal.
Yeah, so I think the preclinical work is supportive of that hypothesis. I think at least in a broader assessment of MAG lipase inhibitors and their potential to mitigate drug abuse. A lot of work from Aaron Lichtman's lab showing that MAG lipase inhibitors can suppress some of the withdrawal symptoms, for example, of opioid dependence. Cannabis dependence is a little bit harder to model in rodents, but it can be modeled, and I think those studies are ongoing. But I do think that's an interesting hypothesis and relates back to what Daniele was talking about, homeostatic balance, that I think trying to re-achieve that in the context of exogenous drug abuse with this system is an intriguing area to pursue.
Yeah.
If we look at the work that Meg Haney has been developing over the years at Columbia University over this, is really fascinating, is that cannabis withdrawal is systematically accompanied by all the things that Phi inhibitors alleviate. There is irritability, there is anxiety, there is depression. All these things are clearly drivers. This negative dysphoria is clearly a driver in the continuing use of cannabis. And the fact that Phi inhibitors have such an opposite effect tallies well with the Cyril de Souza's data that you were referring to before, suggesting in a fairly substantial clinical trial efficacy of the Pfizer compound in CUD, in cannabis use disorder.
Yeah, to follow up on that briefly too. I mean, the origins of that trial could go back to Pfizer's original study on looking at sleep-wake cycles in healthy subjects, where if you take healthy subjects and poke and prod them in a sleep trial, they undergo transient insomnia, not surprisingly, and you can look at the effect of drugs on rectifying that transient insomnia, and that's where the first effects on mitigating these sorts of perturbations in basal sleep-wake cycle was observed. And I think that, so it's in a way, it was somewhat of a replication of that result in the context of marijuana dependence, right, where sleep withdrawal is a major issue.
So we have a question coming through by Twitter. It's a question about CBD affecting THC at CB1. I think it's probably important to consider the concentrations. In the concentrations that people who consume marijuana would be exposed to THC and CBD in its different forms, does CBD ever reduce the psychoactive effects of THC by preventing its interaction at CB1?
Okay. There's clear evidence going back many years that the ratio of CBD to THC affects the psychoactivity of cannabis. I mean, that's very clear data. So yes, how it does, it's not clear. But I think the CBD effects are more interesting when considering CBD as a therapeutic agent, say in Dravet's or something, because normally CBD levels are not very high. Okay. So as most of you know, the breeding of cannabis over the last few years has decreased the amount of CBD, well, increased the amount of THC. But even if you take THC, CBD at relatively equal levels, CBD levels are substantially lower, and Marilyn can address this more. But if you take CBD therapeutically, you can push the levels pretty high in terms of the amounts that are used for Dravet's, because you don't have the psychoactivity. So there, the concentrations in the graph that I showed, which are 50 nanomolar or so, 100 nanomolar, are easily achievable in serum.
So under conditions of marijuana smoking?
So conditions of marijuana smoking, clearly you have effects, whether they're mediated through negative allosteric modulation or serotonin receptors or the 5 dozen other targets of CBD at those concentrations. One can't say.
And the effects are
generally to decrease the strength of the high or more of the negative consequences of the high to drastically oversimplify the data.
Okay, 2 more questions. So, Dr. Piomelli, do you think that an endocannabinoid deficiency could potentially cause ailments in its own right?
Yes.
And often one sees a deficiency, a natural disease which can be associated with a deficient enzyme of some kind. Do we know of any natural diseases that are caused by deficiencies in any of the endocannabinoid synthetic enzymes?
Well, to my knowledge, there are no human knockouts, if you will, of either the major endocannabinoid metabolizing enzymes, biosynthetic degradation, but there is a common polymorphism in FAW that has been linked to the potential for drug abuse as well as to a variety of anxiety-related syndromes. And so I think those studies are now becoming more frequent now that the polymorphism has been identified and prospectively analyzing them in rigorous clinical studies. But certainly that is not a complete disruption of the enzyme. It's a partial disruption, and our understanding of its impact on FAW and endocannabinoid signaling is in early days. But there is at least some human genetic opportunity there to study that arm with that polymorphism. So.
And finally, is there any evidence that agents that are pharmacologically active at CB2 exclusively should be scheduled in any way? Do they have any psychoactive effects that would make them abusable?
Not that I know of. Not quite as short an answer as Daniele, but pretty much.
Great. Well, please join me in thanking our first panel. And David and Aidan, thank you for moderating.